So, good afternoon. If I could ask people to take their seats, and I'm going to welcome you to the Red Talks, a set of 4 Rensselaer research, education, and discovery presentations that epitomize research in the new Polytechnic and how it integrates with the education of our undergraduate and graduate students. Now, my name is Robert Hull. I'm the Senior Associate Vice President for Research at Rensselaer. I'm also a Professor of Materials Science and Engineering, and I'm going to be the Master of Ceremonies for this event. So a brief introduction. Research at Rensselaer integrates the frontiers of engineering, of basic sciences, of data and computational sciences, cognitive and social sciences. And this marriage, this intellectual marriage, if you will, enables us to develop innovative solutions for the challenges, the grand challenges pressing society. So, for example, we have programs which address mitigation of disease, that address development of new clean and renewable energy sources, that address establishing a resilient national and international infrastructure. So to enable these advances, we have many programs, many platforms, for example, including advanced manufacturing technologies that enable new processes and new products. We develop new materials for applications such as advanced computing, new energy sources, or new forms of medicine. We are pioneering new technologies to harness the enormous data stream that comes out of new communications and new control systems and networks. We are also seeking to better understand human cognition, for example, in culture, in communications, and in navigating the ever increasingly complex global business network. So by the numbers, research at Rensselaer comprises over 30 research centers, 1,200 graduate students, countless undergraduate researchers, and about $100 million a year in annual expenditures. But even more importantly, Rensselaer students and faculty are advancing or doing breakthrough research at the frontiers of an incredibly diverse set of fields. So, for example, understanding the form of our planet Earth billions of years ago, developing self-healing batteries, understanding the biological causes and markers of autism, developing new materials and architectures for the next generation of cognitive computers, and unlocking the fundamental molecular mechanisms of photosynthesis, just to give a few examples. So today we're going to hear from 4 pairs of Rensselaer faculty talking about fields as diverse as artificial intelligence, the planetary ecosystem, biology and biomedicine, and the interface between arts, sciences, and engineering. Now, this is going to be a pretty rapid-fire event. I'm going to ask that we hold questions until the end of the set of presentations, and there will also be the opportunity to meet and talk with the speakers at the reception and dinner later today. So then, without further ado, let me introduce the first pair of speakers, Professor Jim Hendler, who is the director of the Rensselaer Institute for Data Exploration and Applications, and Professor Hui Su, who is the director of the joint Rensselaer-IBM initiative, the Center for Immersive Systems, Cognitive and Immersive Systems Laboratory. And they will be giving a presentation titled Putting the Eyes into AI. So thank you, Jim.
So it's my guess that some of you in the audience have heard the term AI lately. It tends to be resonating around the Valley a lot. But it's actually a lot of different things. And so what Wei and I are going to do today is show you some of the exciting things going on in our space, both using the technology, advancing the technology, et cetera. But before we get there, as Robert mentioned, I'm the director of our Institute for Data Exploration Applications. We have a lot of different areas we're working across. Some of you who are over in the exhibition hall before saw some of the many things going on in the campus on life sciences and healthcare, and you'll hear more about that in a talk later. we're doing things in business, we're doing things, of course, many things, you'll hear more about the work we're doing both in environmental and data-driven physical sciences. But today we're really going to focus in this talk on the machine learning and artificial intelligence. And again, as I said before, those are terms people use a lot. But what do they really mean? And so let's look sort of as we look up the sort of tree of life. What is this stuff we call intelligence? So when we start, right, when we look at little things that are in a petri dish or clams or things like that, we don't tend to think of those as very intelligent. But at least they're reacting to what's going on in their environment. As you go a little higher up, you get things that are pursuing and avoiding. They can manipulate the environment to some reason. So these things are essentially embedded or immersed in their environments. But where things really start to get interesting from the AI point of view is when we start moving up to a couple different things. One of them is interaction, right? So if you look at cats with their owners or a team of dogs or wolves cooperating, you start to see that the individual is a different thing than the group. So the first I in these AIs is going to be interaction. The second one, and the one we really start talking about intelligence usually, is intention. So things that are doing things for a purpose, and where we feel like that purpose involves problem solving, making things happen, et cetera. So when we start talking about chimps and dolphins, things like that, we start to see a very different kind of behavior than we saw before. But when we go up to college students and other intelligent entities, there's this big jump, right? There's this very big difference between sort of what we can do as humans and what these other entities can do. And so what we've been looking at, and when we start talking about artificial intelligence, is really across this whole spectrum. In fact, in past research, a famous paper in the 1970s by Howard Gardner looked at a lot of different kinds of intelligence. This was mostly in the context of learning, and there were other ways to break it down. But what you're going to see as we talk about what we're doing today, we're looking at learning, we're looking at intelligence that has to do with being in your environment, how you interact with other people, how you know what's going on. You'll hear later talk about what we might call musical intelligence, and then sort of the stuff we tend to think about, the logical, mathematical. And the question becomes, where does the computer fit into all this, right? How do these different eyes we've been talking about impact what we can do now in the computational space, right?
Okay, thank you, Jim. Following what Jim said about the evolution of the intelligence, I'm going to talk about how humans and AI collaborate with each other. I'm going to talk about a new initiative called Cognitive and Immersive System Lab at MPAC in this beautiful building, Curtis R. Priam Experimental Media and Performing Arts Center in Rensselaer Polytechnic Institute. The research we are working on is how we can augment group intelligence via human-scale immersive spaces. Let me tell you why. Traditionally, AI and humans are interacting in an individual way. So each individuals collaborate with AIs, no matter in your computers or in the cloud, through the traditional human-computer interactions. And when people meet together, like when we meet together, we are making decisions, learning together, have a conversation together. AI is not there listening to what we do and also joining our discussion. It's not there yet. So the group activity is often separated from individuals' human-computer interaction. What really need to be is we want when human and human are interacting with each other, human and AIs are interacting at the same time, and AI are interacting at the same time, so that they can really blend together. The reason why we do this is because we want to immerse AI into the human cognitive processes at a real time. It's not offline. So in our conversation, AI should contribute. And also, we want to immerse human cognitive process into the bigger data and more sophisticated logics. So we call this cognitive immersion, the first I, because our talk is talk about how to put eyes into the AI. So we are going to talk about what we do in research. to augment group intelligence. So in order to achieve the cognitive immersion, we first need to think about how we build up the cognitive and immersive environment. Imagine a group of people are sitting in this immersive environment. For the AI behind the scene to talk to these people, you have to build a dialogue loop for the group of people to be able to talk to the society of cognitive agents or society of the AIs. So we need to think about how the room can perceive what's happening in the human activity in the physical space, understand and reason about human intention, and then collaborate with human beings, and finally generate stories to tell people something that we don't know. So with that, let's look at each individual areas, what we are working on as a fundamental research. So from this page, you can see we are capturing interaction data from multiple channels. The first technology I'm going to introduce is the meeting analysis technologies we developed, built on top of the natural language understanding and speech recognition technology. So the real-time analysis technology will be able to understand what this person talk about, how many statements he make, how many actions, how many questions. And then we also leverage the sensor technology to understand how many people, when you have multiple people in the room, where they are, who they are, when they speak. And then with the computer vision technology, we'll be able to understand the gestures and where they look at. There's a technology to understand where this girl is looking at on the panoramic display in order to tell the attention she has. So with this rich interaction transcription, the next step, we are able to understand the intention, the second eye or the third eye we are talking about here. So first of all, when two people or three people are meeting together, when one person is saying something, the other person is saying yes, and the third person is nodding their heads. So you can combine these verbal and nonverbal information together, understand, oh yes, they are having an agreement about something. So there's this kind of a moment that machine will be able to understand. And then we also create technology to support people make decisions, for example, the selection process. And also we are building machine learning technologies to understand how people come up with that conclusion in the selection process in order to understand if human beings have some bias for something or what's their preferences. And lastly, but not the least in this area, we are building technologies to reason what will be the next step for this meeting when we have a discussion, to predict what will happen based on our capture of humans' belief in this discussion. So the other area that I'm going to talk about is how a machine, if the machine come up with something, if the machine can tell us who are in the room something that we don't know in the most natural way. So we create an agent to come up with immersive narrative automatically, leveraging the relationship among information nodes, hierarchical, spatial, and temporal relationship among the information nodes, and generate multiple interweaving storylines to tell a story, and even generate analogy when the data points are similar in structures. And then we can generate this data point is very similar to that one because they are structurally similar to each other. And so based on all these, the AI will be able to generate a story in this immersive environment. And actually, this example was a scenario we use our narrative generation technology to generate a story about RPI, about people in the history of RPI and about the event and places and the linkages among them. This is a collaboration with our library there. So as you can see, we do fundamental research in these several areas. What we are really trying to do is to build up this dialogue. Why? We want to enable situations room. The cognitive and situation room is an exemplar of a cognitive and immersive systems. And the reason why we call it a situations room is because we are building several rooms in real-world use cases on the same infrastructure. Let me explain to you the very first one, the cognitive and immersive classroom. So the cognitive and immersive classroom is built for language teaching as a use case for now. We call this project called the Mentoring Project. RPI have built a virtual reality environment to allow students to play a game in a culture-rich environment like a tea house. And in CISO research, we also build, this is actually in Studio 2 of MPAC, we build a virtual restaurant environment in the 360 panoramic display. And then this student, Rahul, who is actually a Chinese 1 student who was just exposed for Chinese for just 1 semester, he's able to talk to the agent over there and complete the food ordering and drink ordering using multimodal interaction. And the agent will be able to do more like, assess what he has done or tell a story about something that he doesn't know. And then the other thing the agent can do is to visualize pitch, tone, contour. Chinese is a tonal language. And then the agent will be able to visualize the native speaker's tone, contour, and also compare native speaker's pitch, tone, contour with what Rahul just said in order to visualize the difference so that Rahul can learn more. And in addition to these, we are building airport environment. You see we put real avatars over there. And we are, as for now, we have a group of 30 students working on street environment, gardening environment, so that we are really integrating 3 things together: gamification, immersion, interaction with the AI agent together, so that the student, before they go to the real China environment, they will experience a lot more than in the traditional classroom. Beyond this, we are building other use cases. This is a cognitive boardroom environment for executives to make a decision, for example, mergers and acquisition. And recently, we get more requirement from companies about other business intelligence, BI scenarios. So this environment will be able to support a lot of the future BIs while applying AI to make the business intelligence process to be very different. We are also building healthcare environment for diagnostic scenarios. We are also, the last one is we are building intelligent analyst room to allow several intelligent analysts to make sense of some critical information about a critical situation. And the AI will be able to help the agent to suggest what's going to be the next actions that they can come up with to make suggestions to the government in some critical situations as well. So with that, I will give it back to Jim to talk about the other I's in this talk.
So one of the reasons you've been hearing so much about AI is that a lot of new technologies have really come around a leg in the curve. But there's this tendency to kind of think we just use these things, we put them together, and it's, what you might say is yabba dabba do time. It's just time to get it all together and run. But there's still a lot of hard challenges. So these are some of the things that are out there right now. So there was the Learning by Association was actually what powered the Jeopardy program that IBM built, run by, of course, a RPI alum who was the head of the team, as well as reporting up to John Kelly, who is a member of our board and also an alum. We have work going on in deep learning and neural networks, autonomous vehicles, and learning from human behaviors. And I'd love to have hours to talk about this as I do in my AI course. But the interesting thing about this is all of this is powering this revolutionary stuff, but it's still very limited. And if we're going to do the kinds of environments that Wei was talking about, we have to go beyond. So if we look at just this one little example from some of the things I was showing you, nowadays computers can be trained to do really nice things in vision. So here's recognizing this is a dog, this is a person, this is a chair, and we can answer questions like, which would you sit in? Hopefully you all guess the chair. Which can bite what? Dog could bite the kid could bite the dog. If you think about it a little harder, you'll realize the kid and the dog can both bite the chair, but you wouldn't think that the chair would bite the dog or the kid, right? Or which could be a computer scientist. You need a little bit of problem solving to realize that a computer scientist is a person, the baby is a person. Okay, so that one could do it. But imagine you ask this question, right? Which would you save if the house was on fire? Now, most of you, I hope, are thinking, I would feel bad about the dog, I would save the kid, and I don't care much about the chair, right? And the question becomes, would you use a robot babysitter without knowing which of those it was going to decide? So we don't want some kind of trained system that has a good probability of maybe doing the right thing. We want something that really knows. And so my colleague Selma Bringstorff is working on ethics in robotics. How do we bring those kind of things together so, again, we can have provable bounds on their behaviors? Or here's another example. If I show you this picture and say, what is the relationship between this man and this woman, this is something that's still a little bit challenging to current AI systems. But for example, one of our PhD students is finishing up his doctorate in part on this problem, where he can get something like using, this is what's called a generative adversarial network, for those of you who are into these things, can actually say, so we say a woman who's wearing a blue dress is next to a man whose shirt is white, and doesn't quite get the dancing, but it's getting there, okay? But if I actually show you this, most of you would now have a lot more potential information, because just seeing this woman in the white shirt, white dress in the background tells you it's probably a wedding. If this is a wedding, that might be the groom dancing with his mother, right? That's a fairly standard wedding thing. On the other hand, if I gave you more information, if I told you the groom is an orphan, you'd say, okay, well, that can't be the mother, must be something else. So we can't just use computer training and just keep giving it more and more complex information, because eventually you have to give it the whole world. So somehow this bringing the knowledge into the thing is really crucial. And that's a big area of our research. And it's, it's easy to talk about something like the wedding thing, but here's a real world example, right? This is a CT scan of somebody, and we're trying to say, is this a cancer tumor over here? Now, somebody who knows something about cancer thing would see this unbalanced thing, would actually think, yes, this is probably a tumor. But in fact, if you pulled up the if you pulled up the medical record for this patient, and you discovered that the patient actually has had a mastectomy, then that explains the imbalance, and it's probably not a tumor. So again, you have to use this different knowledge than the knowledge about the picture to be able to make these decisions. So one of the things we're working very hard on at Rensselaer is how do we do AI and machine learning for healthcare, bio, et cetera, and you'll hear a little more about some of that later. We're also looking at a number of the other global challenges. I could tell similar stories in all of these areas. So cybersecurity, a major challenge. How do we look at things like insider threat? The Internet of Things and edge computing is driving us towards many more things, very complex reasoning. AI in the arts, you'll hear a little bit about later. The healthcare I already mentioned, the robotic ethics. And a big thing we're doing is a lot of work, particularly in the Jefferson Project, looking at environmental science. How do we apply these things to solving the problems of keeping water clean, keeping people healthy, et cetera? So I'm going to finish by just saying what I hope we've shown you is that we're working on a lot of different aspects of this artificial intelligence problem. Immersion, interaction, attention, and this more general intelligence. And you can't see it here, but it says, what every good talk at Rensselaer should end at, why not change the world? Because these are technologies that will help us do it. Thank you.
So our next presentation is going to be by Professors Karyn Rogers and Peter Fox, who are both professors in the Earth and Environmental Sciences Department, and they're giving a presentation titled, "The Earth is Telling Us Something, Will We Listen?"
Thank you. So the planet is telling us something. Are we going to listen?
We have to listen. I'm Peter Fox. I'm a Constellation Chair in the Tetherless World. And apart from being in Earth and Environmental Science, I also have an appointment in Computer Science and Cognitive Science. And I have a lot of jobs on campus. But one of the fun jobs is this one. And my view of listening to the Earth is a data-driven and highly analytics-focused approach. And we're going to tell you about that today.
I'm pretty sure that makes me Karyn Rogers. I am in Earth and Environmental Sciences. I'm also the director of the New York Center for Astrobiology at RPI. And I wear several hats. I'm a geochemist, I'm a geomicrobiologist, I'm an astrobiologist. I spend a lot of time thinking about this planet in its infancy and the origins of life on this planet. And so part of my day job, and my night job, and my weekend job sometimes, is to find better ways to listen to the planet, but also teach our students, both in the classroom and in my lab, how they can better listen to the planet as well. And so as part of this job of mine, I was in San Diego last year, and I was giving a talk at an Origins of Life conference, and I was describing to my colleagues what the early Earth was like. And I started going through this long list of characteristics, and my colleague Niles Lehman stopped me and he said, "Karyn, wait, stop right there. I don't understand. That is not the early Earth that I was taught. Those are sort of not the descriptors that I came up with." You see, when Niles was sort of learning about the origins of life in the early Earth, he remembered the famous Miller-Urey spark discharge experiments. And those were very different conditions from what we know about the early Earth today. And so Niles stopped me. He said, "Karyn, I don't understand. What you're talking about is different than what I learned. So what happened?" And I sort of sheepishly said, "Well, Niles, we learned things in those last 70 years. And so in the next 12 or 15 minutes, we're going to tell you about some of those things we've learned. We're going to take you through everything from the origin of the planet to the origin of life. We're going to cover 4.5 billion years of history. We're going to cover 27 orders of magnitude in spatial scales. And we're going to start with the very smallest spatial scale, which are these microbes and the origin of life." So traditionally, when people study the origin of life, they start with life as we know it. They start with these microbes. They break those microbes down into parts, their metabolisms, their biomolecules, and they try and create those parts in the laboratory. And then we go try and find those laboratory conditions on the early Earth. The problem is that that doesn't usually work because our laboratory conditions rarely look like the early Earth. And so this is a problem if you're trying to understand the origin of life. And so what the astrobiology team at RPI is doing instead is we're starting with the Earth first. And so we're starting with this Earth first paradigm. And in this scenario, what we do is we understand first what the early Earth looked like, what all the characteristics are of each of those early Earth environments, and we build those environments in the lab. And we know those had to exist. And so whatever comes out of those laboratory experiments tells us what could have actually happened on the planet itself as life arose. And eventually, we will be able to find an origin of life that could have truly happened on this planet. And so if you're going to start with this Earth first paradigm, you need to know something about the early Earth. You might have the question, was there water? What was in the atmosphere? Was there land? Those are all very good questions, and when I had those questions, I'm very lucky at RPI. I just walked down the hall and see my colleague, Bruce Watson. He's an Institute Professor for Science. He's a member of the National Academy of Sciences, and he is one of the very few people in the world who can ask the early Earth question and get answers. So Bruce has spent decades of his career trying to get these zircon minerals to talk to him. These zircon minerals date back 4.4 billion years, and after a number of experiments over decades and decades, what Bruce has learned is that there was actually water on the surface of our planet within a mere 150 million years of our planet forming, solidifying. In addition, he has determined that the atmosphere in the first 500 million years was likely carbon dioxide and nitrogen. And he's learned a number of these things. Also that the land, with the sort of early emerging continents, actually had rocks that looked very similar to the rocks I grew up with in New Hampshire. So from that, we can start building a picture of what the planet was like. But of course, we know that the planet's not like that now, and so it must have changed over time. Over 4.5 billion years of Earth's history, the continents have sort of moved about the surface of the planet as the plates moved. Ocean basins have opened and closed, and one of the more notable changes was the change in our atmosphere. So the rise of oxygen at 2.5 billion years ago was brought on by life and is one of the most significant changes that we've seen. But it's not the only change we've seen.
It's not. And in fact, minerals provide a key indicator of what has gone on in the planet, in the atmosphere, on the surface over those billions of years. And so, zircons are just but one mineral. There are 5,200 minerals, approximately known, mineral species. They were formed by chemistry, they were formed by biology. And we can study these grouped by the characteristic signature of the metals that are contained in the minerals themselves. Now, of course, some of you are wearing minerals today, a little bit polished. We call them gemstones. These are more, minerals in their raw form, and you can see some examples on the side of the chart there. Now, one thing that we've done, which is, I think, revolutionizing the field of mineralogy, is instead of looking at two-dimensional plots, look, minerals and their abundance over time, we're actually taking a 17-dimensional data approach to these mineral data sets, and we're using social network analysis to study them. In fact, we found out that minerals have a social life.
Minerals have a better social life than some of us, but don't tell anyone.
And so we're using these networks, and what's displayed here across the top at various epochs along the history of Earth in billions of years here is the evolution of a bipartite network of chromium-forming minerals. And what you can see is you go from 4 billion years ago to the modern day, the density of the network changes, the layout of the network changes, the structure changes, and in fact more things we can characterize within that network, not just the clustering of the minerals, based on location, based on similar formation properties, and based on similar compositions, and so on. We can find out a lot of other things, and we can go across all the mineral species types, and we can go to Mars, we can go to the moon, we can go to the asteroids, and fully explore our solar system and look at the changes in the network structures and quantify those changes. And so we're doing that. Now, when you do these network analyses, the first thing you find is that you can make predictions. And when you get these distributions, suggested distribution, large number of rare events, we've recently predicted that there are 1,500 mineral species that we haven't found. Now, we can debate about that. That'd be a good question for later if you want one. And we said, all right, let's go find some. So we started a carbon mineral challenge, and we found 14 new carbon minerals in the last year and a half. And they're being catalogued and named. And what's really key about the minerals is abundance, location, and this availability of oxygen to increase the oxidation state of the elements that are in the minerals. So here's a skyline diagram, and when I said I take a data-oriented approach, whenever we get data, we find ways to visualize and characterize all of the data that's available. So here's 4 billion years along the left diagonal axis, and along the right are typical metals. And this is the availability. This is the mineral locality occurrence by age. And what you should be looking at now is as we march across this skyline diagram, you match it up to when the oceans are opening and closing, the continents are moving, and so metals become more available, they become less available, and the oxidation state changes. So there's more colors in the bars as you go along. And you show this to the mineralogists, and they just go, oh, geez, I've never been able to see this data at all before together in my life. Now, it turns out that in particular, as we start to now look into the biomolecules and the biosphere, this metal availability fundamentally changes the nature of the biosphere. And so we're going to look a little bit at iron, magnesium, and copper. And so here's the diagram in the left, and in the top, well, guess what? A social network of proteins. And these are contemporary proteins. And the layout of the network has data underneath it, so these are not artificially grouped together. And it turns out that the colors here correspond to iron-based proteins, manganese-based proteins, and copper-based proteins. Now, that's informing us today because this protein is for this contemporary period. So what we're doing is studying things in great detail that we know how to do today, and then say, well, what would it look like back in time? Because we know the metal availability, and we can start to run scenarios and speculate about what proteins might have been available in earlier times, and thus start to get to the origins of life. To do this, to make this leap between the short periods that we know in proteins and the longer periods over which we have minerals, a natural place to go is the early life or the recent life. So the fossil record going back 541 million years is a period of study. What's in the top here, and there's a little inset to show you how the oxygen was varying. This is a classic paleontological view. This is their two-dimensional view. They have time on one axis, they have total diversity on another axis, and I'm going to tell you what the colors mean in a bit, but it's punctuated evolution of life. Certain species grow, something happens, maybe an extinction from a comet impact. It could happen because of tectonic forces, non-tectonic forces, climate change, radiation events, diversification are coming and going. And so you look at this diagram, you say, well, that's pretty cool. I can notice the bumps, dips, and wiggles. But if you now do, guess what, a network analysis on this, you get a network diagram that looks like in the left corner here. And you see, again, some traditional clumping together. But if you now go and interrogate the properties of these clusters, then what you find, the pink one corresponds to the Cambrian era. The yellow one corresponds to the Paleozoic era, and the blue one corresponds to the Mesozoic era. So we're extracting out of the structure of the network that time evolution. And what's between those pink, yellow, and blue are great distances. And those great distances are the mass extinctions. And so you can validate these in the paleontological record. But what gets actually really much more interesting is within these clusters, you can notice there's even more structure. So there are minor events. So for example, one that you may have heard of, back in 65 million years ago, the poor old dinosaurs bit the dust. And this shows up as a separate part of the network, separated within that major cluster for the Mesozoic. Now,
the extinction of the dinosaurs is an extinction event that we sort of all know about, but there are all sorts of other extinction events that are sort of hidden in this paleontological record, but we can see in the network analysis. In fact, there was a minor extinction 56 million years ago, just 10 million years after the dinosaurs went extinct. And we identified this extinction event with this network analysis, but up until recently, we didn't actually know what caused this extinction event. However, my colleague Morgan Schaller has found 4 different locations on the East Coast that actually have a record of this time period of Earth's history. And as Morgan went into the rock record, he identified a number of items in the rock record. The first were these impact spherules. And what these impact spherules suggest is that this was a comet impact that was likely the cause of this minor extinction. Associated with these impact spherules is this fossil charcoal, which suggests that there were global wildfires that followed this impact. Using some very innovative analytical techniques that Morgan has developed in his lab, what he's learned is that during this period of time right after the impact event, CO2 rose in the atmosphere so that it doubled within just a few hundred years. The global temperature went up by about 5 degrees Celsius, and oceans became acidified. Now, if those things sound familiar, they should, because those are sorts of the orders of magnitude of change that we're seeing today. And so if we want to know how the Earth is going to react to global change, all we have to do is look into the rock record and see how it changed in the past. And at least for this event, we know that it changed in the past by having this rather significant extinction event. Now, what we've shown you so far is that by using this Earth-first paradigm, we can have the Earth talk to us about things. And what we've learned is that the planet and the biosphere sort of evolve in lockstep. But not every single scenario is captured in the record. And so what we might be interested in doing is trying to really ask some specific questions about cause and effect between the planet and the biosphere. So for instance, I'm very interested in microbes, and so what I like to do is I like to ask microbes questions about how they might respond to changes in planetary conditions. Now, you might be surprised to know that most of the microbes on this planet today grow under high-pressure environments. They grow somewhere between 100 and 1,000 times surface pressures. That's how they grow. So if I want to ask these microbes questions, normally I would bring them into the lab, and using traditional methods, if I tried to grow them in the lab and I didn't replicate that high pressure, what I found is that those microbes generally died. Not just dead men or dead people, but dead microbes also can't speak. So they can't answer my questions. And so what we did in my lab, my students and I, is we built this new high-pressure microbial cultivation device. This is the pressurized underwater sample handler. It allows us to grow microbes at pressure. And when we used this to grow microbes, what we found is that those microbes didn't die, they actually grew. And this allowed me to ask them questions. Now I'm taking this device to the bottom of the ocean, to 5,000 meters below the surface of the ocean just next year, in hopes of finding new high-pressure microbes. But in the meantime, what we have learned is that the push, if nothing else, prevents death. The next time I develop a high-pressure piece of equipment, I'm going to work on both preventing death and taxes, but this was a good start. So I think in the last several minutes, we've helped bring Earth into focus just a little bit and walked you through these 4.5 billion years.
So there's the tagline, the punchline. We have fundamentally changed the way we have listened to the planet. We've fundamentally changed the way that we're approaching the grand problems of the origin of life and the evolution of the planet and how it responds. And
now we've got more questions. Thank you.
Thank you.
So our third presentation is by Professors Cynthia Collins and John Dordick, who are both professors of chemical and biological engineering. And John also moonlights as our vice president for research. And their presentation is titled, Humanity's Future Rests with Harnessing Biology's Complexity.
Okay, well, she's Cynthia, I'm John. That's obvious too. And we're going to talk about some of the very exciting work being done in this beautiful facility, our Center for Biotechnology and Interdisciplinary Studies, and talking about how we can take advantage of biology and the complexity of biology and drive our future. And by the way, while we're both chemical engineers, we both started our careers as biologists. So this is kind of an interesting joining of our backgrounds. So let's
actually a chemist, but
well,
you know.
I'm actually a biochemist. But so let's take a look at something that I think many of you probably know, that mankind has used biology even before knowing what biology was. And in fact, there are many examples where we take advantage of our environment and use them for a variety of different products. For example, the oils in poison ivy actually can be used to generate the lacquer that's found on so many distinct artifacts and beautiful Asian culture materials. And we know what that's made out of. It's actually made out of these kinds of chemicals, and those chemicals can be used in any number of different ways, including turning them into interesting macromolecular structures that give rise to drug delivery systems and things like that. This kind of innocuous-looking fungus over here is able to generate a molecule that also looks somewhat innocuous, but it turns out that that fungus, while it doesn't care about somebody having high cholesterol, that various pharmaceutical companies found that that molecule does have the ability to reduce cholesterol levels in your body, and so it can be commercialized. So we've been able to take advantage of what nature can provide to us, even under conditions where nature doesn't want to give us that information very quickly. And here are another set of examples where we are exploiting the biological systems in any number of degrees of complexity to generate interesting products, from biofuels that come from the starch, or more increasingly the cellulose from agricultural residues, to biopolymers, bioplastics that can hold any number of different things that we use now routinely, to mining of ores, fertilizers, antibodies for biotherapeutics, to things that you probably drink every day. If you drink too much of it, that's not great either. But those are very large-scale biological processes that we can take advantage of. So John has told us a little bit about some applications and areas we'd like to be able to take advantage of biology, but often we think about biology as these types of networks, these complex networks. And really, if we want to think about engineering, we need a lot more tools than we have at our disposal. It often feels like we're doing something along the lines of trying to apply a wrench to a petri dish. And so it looks something like this. And so one of the challenges, that myself, John, and many others at RPI are taking upon ourselves is to develop new ways to engineer biological systems so we can actually harness this complexity. And so in my group,
we're really fascinated by microbial communities. I've been obsessed with them for a long time, and so if anyone wants to talk about this later, we can. But one way that I like to think about it is, in the 20th century, we could think about one microbe at a time. But really, we have new tools and new insights that indicate that we need to be looking at mixtures of bacteria. And so many of us have probably heard about the microbiome, and we're going to get to that in a minute. But I'm going to start with telling you about a different kind of side of things where we can actually take this complexity and use it to engineer new systems. And so in my group, I'm actually interested in using a mixture of microbes to enable different types of bioprocesses. So making them work together, essentially dividing labor between different species or strains of microbes, right? So we're not all jacks of all trades. If we want to do complex tasks, we assign tasks to different members of our team who have different expertise. And so we're trying to do the same thing, where essentially we assign different chemical reactions or different processes to different organisms and put them together to carry out complex systems. And so things that we need to consider are, how do I make these organisms grow together? And so we can do things like distribute metabolisms and use ecological insights to ask the organisms and tell the organisms to grow together when we want them to. My group has pioneered a large set of engineered cell-cell communication systems where we take advantage of what bacteria use every day to talk to each other. And so we might think of them as individual organisms only out for themselves, but they've evolved these amazing complex systems where they actually talk to each other and are able to conduct very complex behaviors. And so we've actually engineered organisms that don't usually talk to each other so they can coordinate their behavior, grow when we want them to do, and even do things like build networks that behave like Boolean logic gates, like AND gates, where they're incorporating multiple inputs. And then, of course, we want to use reaction pathway engineering, where we add different tasks to the system. And overall, what we really want to do is to be able to take waste products and plug and play these different organisms to make whatever target we're interested in. That might be a biofuel tomorrow. It might be an antibiotic or anything else. And so just an example of what we're doing in my group is we're actually developing communities that consist of two different types of organisms. The first one is a bacillus. Doesn't really matter for this presentation, but the idea is that this organism secretes proteins, and these proteins allow the cells to degrade cellulose, so a plant waste. And those molecules that are freed in this degradation process can be used by a second organism, E. coli or a similar engineerable organism, to produce whatever target we're interested in. And so we're working on fuels, antifungal compounds, and other types of system. And we found that when you put these organisms together in this type of approach, you get unexpected synergies. And so instead of having a single microbe where you're asking them to do everything, we're actually seeing the process goes faster and more efficiently by doing this community-based approach. And so, of course, as I mentioned, you hear about the microbiome in the news every day. And so this is one of the areas where harnessing complexity and thinking about this system and how we interact with the microbes in our body and every day is really important. So I'm going to turn things over to John to tell us a little bit more about we've been working on this area.
So while microbes do a lot of good things, they obviously do some pretty bad things. And nature has been relentless in giving us major problems that we have never really been able to solve. I suppose some people might say we'll never fully solve that, but at the end of the day, we try the best we can to keep up. So whether it be in hospital-acquired infections like methicillin-resistant Staph aureus, or through some sinister things such as developing spores with anthrax, there are things that have dire consequences on us as humans. If you take a look at hospital-acquired infections, just looking at those particularly that are drug-resistant, you can see that we've seen significant mortality, and there's, of course, larger morbidity that occurs with these pathogens. Just one of the more common ones today is Clostridium difficile. Clostridium difficile exists in your body all the time, but when they come out of a typical microbiome that we have, for example, if you use antibiotics, you end up having that microorganism begin to take over. And in fact, the pharmaceutical companies have generally been unable to keep up with the development of new antibiotics, and in many cases don't want to get involved in developing these kinds of molecules, particularly because they're not really for chronic infections, which means that it's often hard to make a whole lot of money. And when they do try to use them, they try to sell as many of them as they can. I'm not trying to attack a pharma company here, but at the end of the day, what you end up with is overuse of antibiotics, particularly against infections where they would have no impact. But look at the societal cost. In the US alone, bacterial infections lead to about $55 billion a year in costs. Forget the lives that it impacts, just the cost. Right now, the NIAID, the Allergy and Infectious Disease Institute, gets about $5 billion budget. So that's 11 times what the US federal government pretty much spends on this. Hospital-acquired infections are now the 4th or 5th leading cause of death in the US. And the World Health Organization has already indicated that we're heading back towards the beginning of the 20th century in terms of our ability to counteract many infections. Many surgeries now don't get done because the surgeries are done fine. The infections can be a problem. So how do we deal with that? Well, really you're looking at nature. How does nature heal itself? And how might we be able to take advantage of it? So if you take a look at when you take an antibiotic, you have a series of good bacteria and bad bacteria in your body. Many times, the bad bacteria are already there. We don't really know why, in most cases, the good bacteria prevent the bad bacteria from being really bad. But what we do know is when the antibiotics are used, you go after all of them. You reduce the level of bacteria in your gut. The bad bacteria actually tend to be a little more resistant, and so they remain at a higher concentration in your gut, and then they take over. And what happens then is you end up with these drug-resistant bacterial infections. So what we want to do is be able to selectively, not completely, but selectively kill the bad bacteria while preserving the good bacteria. And what we do is we look at nature for how to do that. Because if nature didn't have a way to get rid of these bad bacteria, then that's all you'd see on this planet would be the bad bacteria. So clearly nature has a way to do it. And one way to do that is actually take advantage of some unique mechanisms, unique biochemistries that exist within the viruses of the bacteria, we call bacteriophages, that can very specifically and selectively kill bacteria. So here, for example, might be a methicillin-resistant Staph aureus, and on the bottom, when it's attacked by these phages, they literally blow up. And I'm not kidding, you can actually see the bacterial cells blow up. And so what we want to do is actually take advantage, and we have begun to do this, take advantage of not the phages themselves, but the enzymes that exist within those phages. And with enzymes, you can do a lot of interesting things. For example, you can begin to mix and match them. You could take parts of one enzyme, parts of another enzyme, bring them together. In fact, they tend to be modular, these kinds of enzymes. And we can incorporate them in any number of different kinds of products. So for example, you can take those enzymes. They're not living. They're actually catalysts. And you can put them into paints and coatings and things like that. And you can then paint the rooms in the hospitals. You can paint the medical or coat the medical instruments. You can even weave them into the bedding and so forth. That can be somewhat antiseptic, but specific to an organism that you don't want to have. So for example, on the right, you see materials that we made. These are paints that used actual human-derived MRSA samples through our collaborations at Albany Med. And you can see that in the white bars, that just means that in the absence of our paint, these bacteria just loved it. They lived perfectly fine, but in the presence of that paint, you get a dramatic drop. 99.9 or 99.99% of those bacteria are killed within just a couple hours.
So another area where we've been looking at microbes has been on the space shuttle. And so we were lucky enough to send some communities of bacteria up on a couple of the last space shuttle missions and wanted to look at how they grow. It's really important to think about microbes and how they grow during spaceflight for human health, right? Here we're able to go out and get antibiotics when we need them. You know, if you're on a long-term mission to Mars or beyond, you are essentially stuck in a small space machine, hopefully taking you where you want to go. And the only microbes in there are from the people, right? You can sterilize those surfaces all you want, but we can't be healthy without our microbes, so we got to take them with us. And so we really need to think about how those microbes are going to adapt during spaceflight and how those might affect, right, long-term crew health. And so what we wanted to look at was the formation of biofilms. So this is a community behavior of microbes. They form on your teeth every morning, right? Everyone knows, right? All that fun stuff. And we were really excited because we saw there was this difference in microbial biofilm formation during space. And so one of the differences we saw was the formation of a really different structure. And so this is just illustrated on that middle panel, where on Earth we see this kind of flat biofilm, whereas during spaceflight we saw these columns forming and a canopy on top of it. And it's actually an adaptation to what we see under hydrodynamic flow on Earth. And we found that this was motility dependent. And so we learned some really cool stuff about the biology of these cells. But we also learned that maybe we need to be thinking about how these microbes might adapt in an unexpected way during spaceflight. And luckily, I was able to collaborate with John on some things. And we were able to look at some of those proteins he just mentioned and how they might be effective during spaceflight.
Right, so lysostaphin, if you remember your Greek, lys meaning to kill, and staphyn meaning Staphylococcus, which is the organism we targeted. And actually, you can see that the killing is better up in space. And one thing that Cynthia didn't mention is this was actually used in the last space shuttle mission that we ever had. And it may be one of the last, if not the last, experiment run, because we had to bring those organisms back and be able to measure what lived and what died. So while NASA won't tell us, we are thinking that it may be one of the last experiments ever run, maybe the last experiment ever run in the US shuttle.
So similar to that environment on the space shuttle, right, we're all spending a lot of time indoors these days. And we're interacting with microbes all the time on surfaces that are coming in through the HVAC systems and windows. And so there's opportunities in that area to think about essentially the environment in the built environment and the microbes of the built environment and how they may be affecting human health long term. Especially in Beijing, in cities like that this is actually an important issue. And also improving indoor air quality. And so in working with architects at the Center for Architecture, Science and Ecology at RPI, we're actually working to build new systems that can harness biological and traditional mechanical aspects in order to improve indoor air quality. And so you can see here you actually have a combination of plants and of course the microbes that are associated with those plants that can improve indoor air quality. And my group has been studying the microbes that are associated with these plants living in this kind of system. And we've been looking specifically whether or not there are specific functions in these systems. And what we're really excited about is we've recently identified the ability to degrade volatile organic compounds. So these are compounds that are in the indoor environment that we might want to essentially both grab and then degrade in this type of system. And so we hope that these kind of new approaches will essentially transform how we think about indoor air quality and mitigating potential infections long term.
So we talked a little bit about bacteria. I'm bringing all the bad news here. Bacteria. What about viruses? And that's a much more difficult problem. I'm not saying bacteria, getting rid of bacteria is easy, but viruses are much more difficult. We know about the impact of this. Every so many years we get influenza outbreaks. The one in 1918 was particularly bad. We don't really know why, although we have some ideas. More recently, Zika, which probably 90% of us in the room here have antibodies against. It doesn't really impact us very much, but at certain points of development of the fetus, it can be devastating. Vaccines, the primary way to deal with them, sometimes they don't work very well. Sometimes we don't hit the right target. There are some universal vaccines that are being developed, but they have a ways to go. We have some small molecules that you can take, and it may mitigate some of the effects of the disease if used early, but not very often. So how do we get rid of viruses? Well, one way to get rid of viruses is to get rid of them from the environment. So if you're going to, and the other way, of course, is as a therapeutic. So in getting rid of it from the environment, here's an example of some work where we took that same paint, but in this case, using a different kind of an enzyme that is able to generate a very highly, but high locally toxic agent, peracetic acid, which we know can kill so many different things, but at very low concentrations in the vicinity of the surface of the paint. That interesting looking enzyme that looks like a donut can actually generate a significant concentration of product that kills viruses. In this case, an influenza A virus, with just in a few minutes, 100% of the virus titer is gone. In addition to that, you can develop some therapeutics. So with Bob Linhart as a Constellation professor in our CBIS, we're looking at various dendrimeric species. These are polymeric species that contain various sugar molecules that bind very tightly, almost like Velcro, to the surface of the virus. And when it does that, it actually prevents the virus from being able to replicate inside the cell. And you can actually see some of the virus particles sitting on top of a cell. And if you take a look at what happens to that poor, unfortunate set of mice, in the absence of this therapeutic, within 10 days, all the mice are dead, but 80% of the mice remain alive for good if given that therapeutic, even after the infection has begun. So if you take a look at where we're going, it's very hard to predict, but we do know the following. Things are accelerating. Our ability to develop new technologies is rapidly expanding. And the ability to go to the next major event in humankind is shrinking very, very rapidly. And this is just an example of a log plot that gives you that information. So there's a lot of things that we are looking forward to help us live better lives, to help us live more productive lives and healthier lives. And all of this really comes through understanding what nature can provide.
And so hopefully, we've given you a good sense of some of the aspects of what's going on at RPI, from biomanufacturing to new strategies to improve human health. And I think the really cool takeaway is that if we stop thinking about complexity as a bad thing and take it and start thinking about what is it we can learn about the context and what will that tell us about where we should go next, whether or not it's combating a disease or making a new product. And so thank you for your time.
Our last presentation is to be given by Professor Curtis Bahn, Professor of Arts, and Professor Jonas Braasch, Professor of Architecture, on the ArtX experience.
Welcome, I'm Jonas Braasch. I'm in the School of Architecture. I'm an acoustician, and my real interests are musical machines, also music practice, and also I'm very interested in architectural spaces and the evolution of how we communicate in architectural scenarios.
Hello, I'm Curtis Bahn. I'm a professor in the arts department, and I teach interdisciplinary classes that bridge arts and sciences. I'm also very active in the ArtX program. As part of President Jackson's vision of the new Polytechnic, you probably know that ArtX is an initiative designed to help Rensselaer students discover the art and science and technology and the science and technology within art. ArtX is the nexus of the intellectual exploration across disciplines, the experience of interdisciplinary teaching. We can trace the sensibility of the ancient concept, or to the ancient concept of the quadrivium. The concept of the quadrivium originated with Pythagoras around 500 BC. The Pythagoreans sought the unchanging fundamentals underlying nature and society, the eternal laws of the universe. They organized their studies into 4 disciplines. First of all, arithmetic, looking into the infinite array of numbers. Second, geometry, moving beyond the line to higher dimensional spaces. Third, to astronomy, the application of geometry to the world of space. And last, music and harmony, which is fundamentally the application of the pure science of mathematics to the evolution of time. It was thought that through the contemplation of the relationships of these essential elements, one could come to an understanding of the essence of humanity. This also could include a view of the relationships between rational and irrational thought.
So we were thinking about looking a little bit into rational thinking and intuitive thinking. So let's focus a little bit about intuitive thinking. So you could think about many processes, especially art processes, are process-driven and intuitively thought. And especially in science, we're often driven by rational thinking, where we have our models, and we try to, with deductive models, try to understand to make sense of data. So I'm kind of proud that my research goes 40,000 years back. I know we heard about research that goes 4 billion years back. And 40,000 years ago, believe it or not, people already built musical instruments, and you find them in cave. This is a flute made out of a vulture bone, and it has tone hole, has finger hole. It's pretty much like a current flute. And you would start to think, well, how would people select the positions of the tone holes that then determine the musical scale that will sound? And if you read the literature, some people think they already knew how to make pentatonic scales, which are the fundamental scales of modern instruments. However, if you look at a current wood flute, all the tone holes are arranged at the lower half of the instrument, and here you have about the octave, while this one is equally spaced. So we think that people intuitively made the tone holes symmetrically, and also so that you can play them with the fingers. The theory to really, the underlying theory really came with Pythagoras 500 before Christ, and then you have the relationship between string lengths and musical intervals. Another really interesting factor is where the flute was found. It was found in a cave. And if you look at the acoustics of a cave, it has 1.9 seconds of reverberation time. That is very similar to modern concert halls. Already 40,000 years ago, people had spaces that sound like a modern hall. And so now you could go about it. How do you understand how people play music? They don't leave music scores for you, right? So you can't read their music. You could try to analyze mathematically how the scale should sound for the flute, or you can do an experimental approach. What we did is, so we built a replica of that flute, arranged the tone holes at the same position, took an impulse response, or the acoustic footprint that we measured in this cave, and then just performed the flute and see experimentally how it could have possibly sounded.
Since the time of Pythagoras and before, in the musical cultures of Europe as well as Asia, mathematical relationships intuitively experienced in the world of sound drove the rational development of scientific systems of pitch, scales, and harmony in music. The most common tuning to our modern ears is the system of equal temperament, where the octave is divided into 12 equal portions. But before the technology existed to tune instruments to this degree of specificity, the well-tempered system developed, allowing for the first time the possibility of working in all 12 major keys and their minor counterpoints. Johann Sebastian Bach was interested in a tuning system that would allow him to explore all 12 keys, and in 1722 wrote his masterpiece, The Well-Tempered Clavier. It was a collection of preludes and fugues in all 24 major and minor keys, in which he explored the musical possibilities inherent in the new matrix of pitches and new instrumental technologies. You can see in the portion of the score at the bottom, the first prelude, thank you, of the Well-Tempered Clavier. Bach's inclusion of sharps and flats indicated the addition of chromatic tones to the standard musical scale and the development of a calculus of chromatic harmony introduced through his work. We can also see the inclusion of a sort of musical and compositional game in which his name, B-A-C-H, was woven into the structure and evolution of the composition.
So one thing we're interested in ArtX is also, can we use some of those art-driven processes to make better engineers and scientists? And if you really look at the history of science, we usually have a lot of rational thinking, and people have models. One of them, for example, is the idea in the 19th century that light must propagate through a medium like sound propagates through air, and that was the famous Michelson-Morley experiment. So Michelson-Morley said, we're going to prove that there is an ether, and what we do is we basically take the motion of the earth that must move through that medium, and then we're going to take a light ray, we're going to divide the light ray with mirrors, and then because relative to the earth they have different speeds moving through that medium, when we bring them back together, we must see interference effects. The problem was they never saw an interference effect. Of course, that's part of your model, because in the 19th century, people knew that this Earth was not the center of the universe. But if you had, back in the pre-Copernicus model, then people would have said, well, we don't see an interference pattern because that proves that the Earth is the center of the universe. And most people argued at the time that the measurement setup was either not precise enough or had systematic errors. But Einstein really was thinking from an intuitive perspective and just said, well, if we can't find a medium or can prove that there is a medium, maybe a medium doesn't exist. And he just did all the mathematics under this assumption, which first led to his special theory of relativity, and later, when he took into account masses and acceleration of masses, also led him to the general theory of relativity. Interestingly, it took many decades before you had proofs, very precise clocks that could measure the effect of the special theory, or later when we had 1916 for the general relativity theory, the proof of gravitational waves, which are basically distortions in the time-space continuum. People believed even before that that Einstein's theory made sense without having any scientific proof. And so we wanted to further elaborate on this idea of intuitive and rational thinking, and we started to build a music agent that can perform live with other musicians. And so basically in this model, we have incorporated the rational thinking with goals and an internal representation of the world. In a musical sense, that internal representation is your music theory, the way scales are arranged, the way rhythms are arranged, but also knowledge that something like a music ensemble exists. But we also have intuitive processes. So these are driven by the actual sound. So what we do is we take microphones that record the musicians live. We have something like an auditory sensor, that's the microphone, and then we perform what we call an auditory scene analysis. So what we do is we basically process the sound with mathematical models in a very similar way to human beings. We extract the pitch, we extract on and offsets of notes and other musical events, we do a timbre analysis and a beat analysis and so on, and that we then use with machine learning tools like neural networks and hidden Markov models to try to find regular patterns that interact with the knowledge and then drive a virtual synthesizer, a musical synthesizer that can respond live to musicians. And one of the most fascinating aspects of this work, and I have to warn you, this is experimental music, so it's not going to sound like Bach, but our former colleague, Polly Oliveros, who unfortunately passed away in 2016 at the age of 84, she was a fantastic accordion improviser. And what this technology enables us now is based on training this agent with her material, that we have some kind of feeling of still being able to perform with her. So in this example, Doug Van Noort on the laptop and myself on the soprano saxophone perform with this agent who basically plays from Pauline's material by responding live to the musicians. And the sound she plays is an accordion. So now you hear the accordion coming in.
This sort of algorithmically generated music composition can be explored by the students through exercises in creative music coding or creative computer coding. For example, Brownian motion, observed in the motion of molecules and the drifting of smoke, a sort of drunken walk through a range of possibilities, can become a powerful tool when applied to rhythms and note selection and computer-aided composition. Here we see a section of code in the Lisp language instructing the computer to select a note from an array of 7 possibilities, following the note-to-note probabilities inherent in Brownian motion. In other words, the note sequencing is not simply random following a uniform distribution of possibilities, but it follows a path with greater coherence and presents a different intuitive effect on the listener. So we'll hear a short section that sort of wanders down. Short random walk there. Visualizing relationships in the mathematics of natural forms can inspire breathtaking computer graphics, art, and computer graphic effects, but can also give us insights into previously unseen structures in medical diagnostics, big data, and global trends. Likewise, the sonification and auditory display of data can allow us to experience repetitions, structures, data anomalies in previously unheard of ways, leading to new insights and methods for computer-human interactions. In this example, we can hear the auditory display of repetitions and patterns inherent in the individual amino acids, nucleotides, nucleotide pairs, and codons of DNA mapped into musical notes and rhythms. This approach can be used to distinguish complex DNA sequence data from other repetitive data.
So we've talked a lot about how we use scientific processes and rational process in music or for sonifying data, but can we also sort of with technology foster intuitive thinking. So one of the examples that we have at Rensselaer, at MPEC, and in other places, we have a lot of virtual reality system. This one is a collaborative space where you can host up to 50 students. One of the things we do is we have a design a class where students in architecture design buildings at human scale. And in this case, we have a student who programmed a CAD model of a cistern that Pauline Oliveris used to perform music in. That's an underwater tank that's empty, and it has a reverberation time of 45 seconds. So once you clap your hands, you hear the sound decay for 45 seconds. Unfortunately, this building is condemned because of off-gassing, so you can no longer perform in there or record in there. So we basically made an acoustic model with ray tracing, so we traced the acoustic rays and then implemented in a real-time system so you can perform in the space as if you were in the physical space. And the other thing what we're using it for is we have, for example, a symposium on assistive technology in art and music every year, and we host it in our creative reality lab so that people with spinal cord injuries can play in spaces where you wouldn't have very difficult access because of a disability. So this one is an excerpt of our recording in the cistern with the 45 seconds of reverb. So since we have the last talk, we thought we'd leave you with a number of questions that arise during our research and that we hope that all our fantastic students will help to solve us, although with the fantastic facilities we have at RPI here, for example, at MPEC Studio 1. So the first question is, if a human cannot perceive a musical phenomenon, does it have artistic merit?
Which has more aesthetic value, the algorithm or the musical output of the algorithm?
Are computer programming and music composition equivalent acts of human creativity?
Will a computer ever produce a musical work that captures the essence of humanity?
Certainly something to think a lot about, and thank you very much.
So I'd like to just leave time for a couple of questions for the audience. We're running a bit late because one technology we will never invent is one to keep faculty members on time. That I can guarantee. But then there will be plenty of time again to follow up with the speakers at the reception at the dinner this evening. But because you've been such a great audience, I've been watching, people have been enthused, interested, engaged, I do just want to make sure we have a couple of questions. So great, one at the front there. Thank you.
I'm curious to a lot of the speakers, what's like the life hack?
Let's pass the microphone.
Yeah. My question to a lot of the speakers is, what's like the life hack? You know, we've got plants with that affect our microbiomes for the positive. For as an example, there's some other areas of health, and so I'm just curious, like, what did you learn that you now implement into your life, or might recommend us who aren't doing the research to just take it?
Great. So we'd ask John and Cynthia, I think, to answer that question. Are you mic'd, or do you need the? Thank you.
So that's a good question. You know, I think one of the things we can do a little bit more is spend time outdoors, actually spend time with plants, but also think about, you know, new ways to bring the outdoors in. You know, there's a lot of really cool ways to bring that, something like a plant wall inside, but you also need to think about different ways to innovate. We don't want soil in those walls, right? That's a great place for fungus to hang out, and that would probably not be great for the indoor built environment. And so we still need to think about engineering the materials and things like that in that system. So that's an example of kind of where we're going with the kind of technology. You know, I think the life hack might be thinking about, you know, not thinking about microbes as bad or good, but trying to think about them in, like, what's the context? You know, maybe don't always use the antibacterial soap because you might end up actually making things more antibiotic resistant. And so there's actually some good microbes on your skin. Still wash your hands. Don't get the wrong idea. Yeah.
Karyn, if they want to add to that answer. Karyn, do you have anything to add? Sorry, then, Cynthia, do you have anything more?
No, I'm good.
Great. Well, thank you. So let's take one last question right from the front here. Thank you.
Well, okay, so there was something 65 million years ago, and there was something 55 million, and then there was some other ones. So are we due? What's going to happen?
Let me take the microphone and pass it back to the people who can answer that one.
So there's a couple of answers to that question. One is when you take a data-driven approach, our ability to predict and detect oncoming, or in fact, currently ongoing extinction events, there's substantial evidence for that, is bound by the data. And it's bound by scientific responsibility and veracity of results. And so we have ideas, but it's a little bit like the volcano prediction thing and the seismic prediction thing. There are significant consequences for what might happen, as well as exactly how many days. Let's see, 17.
42? No, so for better or for worse, extinctions don't happen immediately, and we don't really get to notice them sometimes until after the fact. What the current network analysis suggests is that we're probably in the middle of one, and so the number of days is a negative number. We've already started this sort of new extinction that we're in. That doesn't mean we can't change it, though.
And I'll just finalize by saying one thing I didn't mention is the network techniques allow us to quantify the relative magnitudes and impacts of past extinctions and radiation events. And it's completely changed our understanding of what was important back in the day and potentially what might be important in the future. So it's a much more quantitative approach.
So there is hope, is the answer.
19 days.
19. Okay, it's got better, yeah. So I would just like to conclude then by thanking you as the audience for attending and for your participation. And I'd like to thank our speakers for really personifying the excitement, the passion, and the brilliance that is research at Rensselaer. So thank you all very much.